Vaucheria occurrence. Majority of the species of Vaucheria are either aquatic or terrestrial. The terrestrial species are found upon damp soils and muds in yellowish-green velvet-like carpets. In terrestrial species, the rhizoid-like branches are present at the base of the thallus which attach it to the substratum. The freshwater species are usually found in aerated water. Plant body and cell structure. The thallus consists of branched, coenocytic unseptate and tubular filament. At the base of the thallus a branched rhizoidal system is present attaching thallus to the substratum is known as hapteron. The septation takes place at the time of the formation of reproductive bodies or injury to the alga. The cell wall is thin and consists of pectose. In the center of the tubular thallus, there is a big central vacuole. The cytoplasm is confined to the peripheral region. The numerous discoid chloroplasts are embedded in the cytoplasm towards outside and numerous minute nuclei are found towards the central vacuole. The pyrenoids are absent in chloroplasts. The starch grains are absent. The reserve food material is oil or leucosan. Reproduction. The reproduction takes place by vegetative, asexual and sexual methods. Point 1. Vegetative reproduction. This type of reproduction takes place by accidental breaking of thalli into small fragments. Each fragment of the filament is capable to give rise to a new plant. Point 2. Asexual reproduction. The asexual reproduction takes place by several means, e.g., by zoospores, aplanospores, hypnospores, etc. By zoospores. The multiflagellated zoospores are developed usually in aquatic forms. A single zoospore develops in a single zoosporangium. Process of zoospore formation. Any distal branch of the thallus may convert into a zoosporangium. Much of the food reserves, chloroplasts and nuclei accumulate in the distal end of the branch of thallus. This distal end is comparatively swollen. Very soon a septum appears at the base of this swollen end. The central vacuole disappears, the nuclei and chloroplasts reverse their position, i.e., the chloroplasts shift inwards and nuclei towards periphery just beneath the cell wall. Opposite to each nucleus two flagella are developed. The terminal portion of the zoosporangium softens and a small pore develops. Through this small terminal aperture the zoospore squeezes out and swims freely in the water. The zoospore swims for about 15 to 20 minutes and then settles down to some substratum, withdraws its flagella and secretes a wall around it. Very soon it germinates, giving rise to 2 or 3 tubular outgrowths and a new branched thallus develops. 2. By aplanospores. The aplanospores develop in dry conditions and especially in terrestrial forms. Here the contents of an aplanosporangium develop into a non-motile aplanospore. This aplanospore escapes by the irregular rupture of the aplanosporangium. On the approach of favorable conditions, the aplanospore germinates producing tubular outgrowths. They may germinate after liberation or within the aplanosporangium. 3. Hypnospores and cysts. Sometimes in terrestrial species the segmentation takes place in the tubular branches forming many small compartments. Around the protoplast of each segment, a thick wall develops and they are called hypnospores. The hypnospores either germinate directly producing new thalli or they divide producing thin-walled cyst. Each cyst germinates in a special way. The cyst breaks and a pore develops at one end of it. The protoplast of this comes out in a moboid fashion, becomes rounded and develops into a new thallus. 3. Sexual reproduction. The sexual reproduction is oogamous. About all the species reproduce by this method. All the freshwater species are homothoric. The sexual reproduction takes place in the forms growing in damp soil or still waters. It does not take place in running water species. In the monoecious species the oogonia and antheridia develop on the same thallus and in dioecious species these organs develop on 2 different thalli. Development of antheridium. Mostly the species are protandrous, i.e., antheridium develops first and oogonium afterwards. The antheridia develop on the lateral branches at their ends shortly before the formation of oogonia. The part of the thallus giving rise to antheridium possesses abundance of cytoplasm, chloroplasts and nuclei. In most of the freshwater species, slender hook-like antheridium develops having a pore at its distal end. A septum develops just beneath the curved portion of the antheridium. The nuclei of the antheridium divide mitotically again and again, and around each nucleus cytoplasm is deposited. Each such small bit metamorphoses in a biflagellate antherozoid. These antherozoids are liberated through the apical round opening of the antheridium. Development of oogonium. As mentioned above, the freshwater species are homothaulic and the oogonium develops on the same filament just near the antheridium after some time. At the time of the development of oogonium, a small mass of colorless multinucleate cytoplasm is accumulated in the thallus just near the antheridium. This colorless mass of cytoplasm is called wandoplasm. The wandoplasm moves in the apical part of the oogonium developed by the lateral outgrowth of the filament. Gradually, the oogonium outgrowth becomes globular and sufficiently large in size. A septum develops at the base of the oogonium, which separates the oogonium from the rest of the plant body. A colorless beak-like structure develops at the upper end of the oogonium. A round uninucleate egg develops inside the oogonium. At maturity, the beak gelatinizes and an opening develops. Fertilization. As soon as the antherozoids liberate from the apical opening of the antheridium, they approach the oogonium. Many antherozoids enter through the opening of the oogonium, but only one lucky antherozoid penetrates the egg and fuse with egg to form diploid zygote. The zygote secretes a multi-layered wall around it and called oospore. The oospore is sufficiently rich in oil reserves. Germination of zygote. The thick-walled zygote oospore undergoes a resting period. This resting period may be of several months. On the approach of favorable conditions, the zygote germinates, producing tubular structure which develops into a new thallus.